Welcome back. This is the second part of the lecture on structural DNA nanotechnology. In the previous lecture, we discussed a method, scaffolded DNA origami, that's proven powerful enough to self-assemble DNA strands into objects that are about twice the mass of a ribosome, about 5 megadaltons in size, involving a long single-stranded scaffold that's folded by many short staple strands into the desired object. For the second segment, I'm going to discuss a new method that was just reported in the last year. This is work that's primarily been led by my colleague Peng Yin at Harvard and the Wyss Institute that he calls DNA single-stranded bricks. And it turns out that this method seems to be roughly comparable in its power of self-assembling structures of this kind of complexity. My group assisted in collaboration at the endpoint of moving this into 3 dimensions. I think it's a really interesting method. That's why I'd like to discuss it in this iBio seminar. The scaffolded DNA origami method is analogous to some toys that we might have played with. So this is a DNA snake that you can fold into 3-dimensional structures. Here's another related idea of a snake-like polymer that we can fold into objects. It's also familiar to biologists who think about polypeptide chains that are individual chains that fold up into some kind of 3-dimensional configuration. And in this way, you can achieve almost any shape by having a long polymer folding that up into that shape. However, from a human point of view, there might be a simpler way if that would actually work, and that's using the example of Lego bricks. So if you can imagine that you have a set of bricks that have a stereotyped shape, if we just have a lot of them and we can connect them at different angles, then now you could argue that gives us even more design flexibility in building these large 3-dimensional shapes. You no longer have to worry about the connectivity of the chain going through the object. And what Peng Yin's group has demonstrated is that, in fact, we can do this with DNA. Now, when DNA origami came out, it was a shock to everybody that, wow, we could build these very complex structures, and then it was immediately assumed that the key to success for the method was the fact that you did have this very long strand that keeps all the short strands into order. It was a master template, and if you didn't have that, maybe everything would descend into chaos. And what Peng Yin's group demonstrated is, in fact, that's not so, although we still think that long strand might be helping, but now we know that it's certainly not a necessary component for a successful strategy to build structures of this size. So the first part of this story was developed in Peng Yin's lab, work led by Brian Wei and Mingjie Dai in his lab. And the idea is as follows. So this is, if you remember from the first lecture, we had that idea of a double crossover tile, and we said, oh, if we could only have double crossover tiles of many different sequences and they would actually behave themselves, then we can now make a complex tapestry where every single one of the elements has a unique sequence. And what Brian and his colleagues demonstrated is that they could do that, but with a slightly different motif. The idea is as follows. So you have each one of your bricks or tiles has the same stereotype architecture where it has 4 different domains, and each one of the domains is one turn of a helix, about, let's say, 10 bases long. And it's a flexible polymer, but in the final design tapestry that it's supposed to self-assemble into, each one of those tiles is supposed to adopt a very fixed orientation where it's like a horseshoe. One half of the horseshoe is part of one double helix, and then the other part of the horseshoe is part of a second double helix. And they assemble with each other using the following rule, that if you have, in your solution, you might have domain 1 of one of your bricks or tiles is going to be compatible with domain 3 of another tile, and then domain 2 of one of your tiles is going to be compatible with domain 4 of another one of the tiles. Each one of these tiles in this assembly, in this tapestry, has a unique sequence, and it has 4 unique nearest neighbors. And so if you can design all of these strands with the same overall length and structure, but each one with a different sequence and with the sequence complementarity rules that I just described, it turns out you can make these objects of the size and complexity of a DNA origami. No long strand required. Here's another representation of that motif, abstract representation that more closely resembles a Lego brick. So what we have are, again, 4 domains. We have domain 1, 2, 3, and then 4. And we can see domain 4 of one of these tiles is now interacting with domain 2 of another one of the tiles. And we have the double helices are running from the lower left-hand corner and now up to the upper right. There's a stereospecificity that's indicated by the shape of the key and the hole, so we can hopefully understand that the key and the hole can only interact in one orientation, and that enforces this coplanarity of the tiles. And the actual physical basis, of course, is that each one of those interactions is one complete turn of the double helix that forces it to be coplanar. And once you understand those principles, hopefully you can see that if you had a bunch of these tiles, each with a different sequence, each with those sequence complementarity between plugs and holes that I described, you can now self-assemble very large tapestries in principle where every location in the tapestry is occupied by a unique tile. And what Brian and his colleagues demonstrated was remarkably that this works, no long strand required. So here what they've done is they've self-assembled a structure with something on the order of a few hundred unique tiles, each one with a stereotyped design position within the tapestry, and they found that they could make these in fairly high yield. So on the left we can see an agarose gel where we can monitor roughly the formation of the object. U means unpurified. And we have the initial building blocks on the bottom. You just cook these for a while. Again, you do this annealing profile where you heat to 65 degrees and cool to room temperature over the course of a day or so. And then when you look on a gel after a day, you can see a large fraction of these building blocks have self-assembled into an object of discrete size. Of course, there's some misassemblies as well. That's where the smears are coming from. But then you can now cut out that band from the gel, and then you have a population of molecules that are enriched for the one that you really want. And in their case, they then looked at these objects using atomic force microscopy, and they saw that they were making rectangles of the desired shape and size. And so first of all, it's just amazing that to a lot of us that this works. You just throw all these sequences together. There's no sequence design. All of the plugs and holes were designed using a random sequence generator, and the method just works. One of the remarkable aspects of this method is that you can now generate new structures simply by repipetting the strand sets and leaving out strands. So for example, if we imagine pipetting that rectangle, but we just leave out the strands corresponding to eyes and the mouth, then now we could generate something like this, a smiley face. And one can imagine, again, just having repipetting these strands with different subsets, and you can now generate different shapes in this way. You can either pipette manually, which becomes tedious if you try to build something like the 100 objects that Peng Yin's group demonstrated. What will be more efficient, and which they eventually implemented, is if you have some pipetting robot that actually does all the pipetting for you. So maybe with the standard pipetting robot, to pipette the pools to build 100 different objects might take a couple of days, but then that can be basically unsupervised. And then comes currently a lot of hard work of the imaging. So far, Peng's group and my group, and I'm not aware of any group that has an automated imaging platform for these objects. But after a lot of labor on the atomic force microscope, one can see that something over 95% of the designed objects actually were able to fold as predicted. So we can see different letters, we can see numbers, Chinese characters, emoticons. We can see a journalist, Ed Yang, was inspired. So in Photoshop, he cut out some of these letters and made a message that says Wyss Institute for Biologically Inspired Engineering at Harvard University. So in the future, we'd like to be able to assemble the letters into this kind of arrangement on their own and without the use of Photoshop. But for now, we think it's already in advance that we can at least make the letters. So here's a movie prepared by Gail McGill that illustrates how we imagine the self-assembly might occur. Again, each one of these tiles has 4 nearest neighbors, and at some point it's going to have to nucleate. And then once you form a nucleus, we believe that that will then grow to the larger structure. Actually, we think the key to the success of this method is that we fold it and that we design it in a way that the nucleation is very slow and the growth is very fast. And in that way, it's kind of like population control, that anytime you form a seed, then it's going to have an abundant supply of food or building blocks in order to grow to its full size. I mean, imagine a situation where nucleation was fast and growth was fast. Then you basically get nuclei and seeds forming everywhere, and very quickly you deplete the pool of building blocks. And at that point, you'd be in trouble because a lot of the seeds would have grown into partial structures. In order to complete their growth, because there's no more building blocks, they would have to start cannibalizing each other. So we think that a robust design principle for programmable self-assembly is to try to build your system so that nucleation is slow or controlled. So we can see with DNA origami, we can now envision those long scaffolds as controlled seeds, that if we're adding an excess of the staple strands, then we know the number of seeds is basically the number of those long strands that we're adding. And in that way, you never run out of the building blocks. In this case, with the single-stranded tiles, it's because that nucleation event is slow and the growth is fast. All right, so I was just in the peanut gallery watching this amazing work going on in my colleague Peng Yin's lab. And Yonggang Ke is a postdoctoral fellow in my group. Luvina Ong is a graduate student in Peng Yin's group. And Yonggang and Luvina decided they wanted to collaborate with Peng and extend this into 3 dimensions. So that's the work I'm going to tell you about next. So just like we were able to extend 2-dimensional DNA origami into 3-dimensional solid structures, Yonggang and Luvina were able to do this using single-stranded bricks. It turns out the principle for converting from 2 dimensions to 3 dimensions is extremely simple, in principle, if it works. So on the upper left-hand corner, we have the diagram that I showed you previously with the 2-dimensional single-stranded tiles, where since each one of these plugs and holes is exactly 1 turn of a double helix, that enforces a stereospecific geometry between the tiles such that they're coplanar. But if you think about it, you could get something that's not coplanar just by changing the length of those plugs and holes so that they're no longer integral numbers of turns of the double helix. So for example, here, what Yonggang did was he designed these plugs and holes to be only 8 base pairs instead of 10. And so now 8 base pairs is roughly 3/4 of a turn. And because it's 3/4 of a turn, then the stereospecific interaction between these bricks is now going to form a dihedral angle of 90 degrees. And we illustrate that with the following arrangement of plugs and holes, so that you can see again the key and the keyhole are only going to fit together, making that dihedral angle of 90 degrees. And that's in physical reality enforced by the fact that it's only 3/4 of a turn, 8 base pairs interacting. So now let's go through a thought experiment that further elaborates this idea that this 90-degree dihedral angle allows the self-assembly of a 3-dimensional solid cuboid structure. So imagine that we have in our CAD program a bunch of these single-stranded bricks. And then the first thing we do is we lump together a bunch of these bricks into these planar groupings. And in this representation, the bricks are not actually interacting with each other with any base pairing. We're just grouping them together in our CAD program for explanatory purposes. The next step is we generate another planar grouping of these bricks, where we've now rotated the orientation of the bricks by 90 degrees counterclockwise. So hopefully by looking at the orientation of these keyholes, you can see that we've rotated the orientation of the bricks by 90 degrees counterclockwise. We can now repeat the process, another 90 degrees counterclockwise, another 90 degrees counterclockwise, and then another 90 degrees counterclockwise. Now the next step is we program those plugs and holes to have unique sequence complementarity. So for example, this plug here is going to be complementary with this hole here, et cetera, et cetera. Again, each one of these single-stranded bricks has a unique sequence, has 4 unique nearest neighbors, and has the desired base complementarity between those nearest neighbor domains. And if you do that, hopefully you can see how you could self-assemble these different planes into this cuboid structure. In fact, you'd just be throwing all of those single-stranded bricks together into a pool and having them self-assemble just like before, but now in 3 dimensions. Furthermore, we can abstract this in terms of the design process in terms of a 3-dimensional canvas, 3-dimensional cuboid canvas, where each one of these volume elements, or voxels, is 2.5 nanometers by 2.5 nanometers by 2.5 nanometers. So in this case, the double helices are again running from the lower left-hand corner to the upper right-hand corner. And each one of these, again, it's 8 base pairs that represents one domain from each of those bricks interacting with each other. So in design space, what we do is we start from this three-dimensional cuboid canvas. We start removing voxels until we end up with a three-dimensional object that we want. Then we have a computer program that will compile this abstract voxel element representation into the brick representation. So the program will ask, okay, what series of bricks do I need to remove in order to allow me to remove individual volume elements? Then whatever series of bricks that are remaining for us to pipette, that's now translated into instructions to the pipetting robot, which will then go and pipette subsets of strands corresponding to whatever kind of object that we want to build. And so again, Peng loves the number 100. So Yonggong and Luvina strove to build over 100 different objects, just like before, but now in 3 dimensions. This represents the different designs that can be created. Now we have letters that are in 3-dimensional relief. We have Chinese characters that are inscribed into 3-dimensional blocks. Same thing with numbers. In this row here, it's an interesting representation where now the solid is supposed to represent bricks that we left out of the assembly. And the translucent represents bricks that we left in. So what this means is that this is supposed to self-assemble into a solid object with a completely enclosed cavity that has a toroidal type arrangement. And then there's, so the pipetting was done by a pipetting robot, so just feed the instructions to the robot, come back in 2 days. And again, we don't yet have an automated imaging platform, so then there's a lot of work involved in generating this figure, where we have electron micrographs now of these different objects. These are projection images. For example, here we can see a little spaceship that we were trying to design. Here's an animation from Gail McGill at Harvard Medical School that is illustrating what we think the dynamics of the structure might be. So now I'm going to go through a series of examples of different kinds of structures, just to give you, again, a feeling in the generality. You start from this 3-dimensional canvas. You start removing volume elements, whittle it down until you get the object you want. So here's an object with that cavity on the inside I just mentioned. So now when you image this in transmission electron microscopy, you're going to get projection images. They're kind of like X-rays. So if you look at the particles in different orientations, then you'd expect to see different images. So for example, you expect to see the O if you're looking from the top down. But if you're looking from the side, then you'd actually expect to see something more like this. Here's an object. It's a 3-dimensional smiley face. Again, each one of these volume elements is 2.5 nanometers by 2.5 nanometers by 2.5 nanometers, 8 base pairs. And looking down from the top, we can see the smiley face. Looking from the side, then we see a different kind of image. Here's an object that's designed to form kind of like a six-sided die, except it's a cheating die, and it only has 3 numbers. So it has different crisscrossing channels to the object. And again, depending on which face lands on the grid, you expect to see different images. So 1, 2, 3. All the same object, just landing in different orientations on the grid. Here's an object that, when you look from the top, is supposed to look like the letter B. And when you look from the side, it's supposed to look like the letter A. And again, that's something that we can see. Here's another object, looks like C from the top and D from the side. Here's an object with basically a channel in the top. And if we look from the top, then we can see this characteristic channel pattern. Again, if we look from the side, then we can see we only removed strands for part of the top of the object. The bottom of the object remains solid. For the 2-dimensional structures, Peng's group developed some software that allows them to quickly design any shape they want. So you can start from some kind of image that you upload into the software. The software will do edge detection and then figure out where the boundaries of the object are. And then based on that algorithm, the program can automatically determine which strands to include in the self-assembly, which ones to leave out. For the three-dimensional structures, this is something that's still in progress, but what Yonggong did was he took his favorite three-dimensional rendering program, told it to render this series of volume elements, and then just what he's doing now in real time is he's carving channels into this cuboid structure. So he's just removing channels. And in real time, we can see him create two crisscrossing channels that are orthogonal. And this gives you a feeling that within just minutes, you can now design any structure you want, very much analogous to what a sculptor is doing. But then it's going to take some time for the pipetting robot to pipette all the strands, for the folding to occur, and then for the imaging is going to be somewhat time-consuming until we have an automated platform for that. So just to recap, we have a design phase where we start from our canvas, two-dimensional, three-dimensional canvas. We figure out which of the bricks we want to exclude. That gets converted by software into pipetting instructions to the robot. Robot does its thing, and then we heat up and cool down the strands over the course of a day or longer for the more complicated objects. And then we look at them using atomic force microscopy or transmission electron microscopy. It's always nice to have some more movies so we can see the pipetting robot in action. And we can envision, hopefully, a day not too far from now where we have everything is automated. So we can just design the objects, and then everything else will be handed automatically, including the imaging. Might make a wonderful resource for students that if they can go online, submit their designs online, and then maybe there's a chance that a lab will actually build the object in the laboratory, and then the student can see their object, an electron micrograph or atomic force micrograph of the object they designed. To summarize up to this point, now we have a second method that allows us to generate objects that are roughly twice the mass of a ribosome or larger. That was just published in the last year from Peng Yin's lab, DNA tiles and bricks now, that doesn't require a long single strand. And for some applications, you could imagine with this overlapping capability, you could arbitrarily choose which one you want to select. However, when we look closer, we could imagine that the independent methods might have different advantages depending on the application. So for example, with DNA origami, we've noticed so far that the assemblies seem to be faster. So although that long strand doesn't seem to be absolutely necessary, we can imagine it does help to speed things up by grabbing the individual strands and bringing them together more quickly. A second advantage is that we believe that the DNA origami, at least as currently constituted, could be thermodynamically more stable to have this long strand running through the entire object. We can imagine the thought experiment of what if we started from the DNA tiles and then just started ligating some of those tiles or bricks together to make a long strand. Then it should be more stable. So in that way, we imagine DNA origami has more linkages between the strands, longer strand, then it should be more stable, at least currently. And finally, we can imagine that DNA origami probably can offer greater mechanical strength, that if you have that long scaffold strand is crisscrossing throughout the entire structure, now you have to break covalent bonds, probably, in order to really disrupt the object. Whereas with the DNA tile object, now if you can imagine creating a facet of breakage without having to sever any covalent bonds. So what are the potential advantages of DNA tiles or bricks over origami? Well, one is that the design is more modular. It corresponds more to our intuition of how Lego bricks can be designed. It's conceptually simpler, and that's usually something that is desirable. Oftentimes when the design process is simpler, then it's going to be more versatile and more powerful. It'll be better for teaching to students how this works. And then finally, the DNA tiles offer the advantage of synthetic diversity, because all of these elements are short strands, then they're accessible through synthetic chemistry, which means we can put any kind of nucleoside analog that we want in there, assuming it's still base pairs. Whereas with the DNA origami, because it's relying on this long single strand, currently our only way to generate these very long single strands is enzymatically. And therefore, we're limited to those nucleoside triphosphates that are recognized by DNA polymerases. So where could this potentially be advantageous? So let's say that you're trying to self-assemble a drug delivery vehicle. Maybe if you built it with DNA origami, you'd start to worry about, well, maybe nucleases are going to digest my long strand. Maybe my long strand is going to trigger an innate immune response. But if that's my concern, then maybe I should think about designing the same kind of structure but with DNA bricks, where I can use, let's say, mirror image building blocks that are nuclease resistant and that are not recognized by the innate immune response. What we found is that, again, for these discrete objects, maybe the performance of the two methods is similar, but where the DNA brick method really seems to shine is in building periodic structures. So what we've done here is, what Yonggang has done is he's programmed the right-hand side of this lightly shaded unit cell to have complementary sticky ends to the left-hand side, or complementary plugs and holes, I should say, and complementary plugs and holes from the front end and the back end. And so now what will happen is that that unit cell won't stop with a discrete object. It'll actually polymerize into a two-dimensional lattice. Furthermore, we don't think that it's forming hierarchically. It's not that you form a bunch of unit cells and then those unit cells assemble. Rather, we believe that the assembly is growing piece by piece. So each individual brick is adding on one by one. And in that way, looking at this periodic assembly, actually, if you think about it, a thought experiment, the definition of the unit cell now is a little bit arbitrary because we could just as easily draw a unit cell connecting these four corners. It's equivalent with these periodic structures. Anyways, the important thing is that this single-stranded brick method seems to give us better performance in the test tube in making these periodic structures. So this is a quite remarkable design that was developed by Yonggang, where the helices are pointing up out of the plane of the DNA crystal, and the unit cell has dimensions of 6 helices by 6 helices, so about 15 nanometers by 15 nanometers. And in this particular example, he designed a cavity within the unit cell of a 2 by 2 helices that are missing. And then what this is going to do is now self-assemble into a crystal that where, again, the unit cell has dimensions of about 15 nanometers, the hole's dimensions about 5 nanometers, and the entire crystal can grow to multiple microns in dimension. We believe that these kinds of structures could have application as templates for perhaps growing inorganic materials and to make molecular wires and plasmonic devices. We think that it might also have application in biology for something like the host-guest crystallography that Ned Seaman envisioned. In this case, in this example, it would be two-dimensional. So what if we could get membrane proteins to assemble into stereotyped orientations and locations within these cavities and use the DNA crystal in order to impose that crystalline order on those proteins? That could be a way to accelerate structural biology research. So this is just some more examples of periodic two-dimensional crystals. In this case, what Yonggang is doing is he's polymerizing in the direction of the helices. So again, every cylinder is a double helix. And we can see these precise channels. It's the same story as with the discrete objects. He just starts from a solid cuboid unit cell and then removes strands in order to create the cavity features. And in this way, it can create an extremely diverse set of crystals with intricate features that is basically not accessible using any other known method. So this is an interesting example where what he did was he made a very thin crystal that was only, I believe, 32 base pairs in height. And now it turned out with this design, the structure no longer wanted to be planar, but instead had a tendency to wrap around to make a tube. And we can see these nanotubes that have an appearance that's somewhat reminiscent of biological assemblies, such as this is a tobacco mosaic virus. Of course, this object is made entirely out of DNA. It's not infective. Yonggang and Wei Sun in Peng Yin's lab have gone on to use these crystals to template the self-assembly of gold nanoparticles onto them, again, potentially for electronics or photonics type applications. And so what they've done here is they've decorated 5 nanometer gold nanoparticles with single-stranded glue, and then they have the complementary glue that's lining the inside of these channels. And in that way, they're able to get high densities of these gold particles into those channels. Here, what they've done is they've just coated the entire surface with a high density of these 5 nanometer gold nanoparticles. I should mention that although DNA origami is not as good as DNA bricks for making these two-dimensional structures, it does have some ability to do that. So this is work that from Yonggang that we didn't publish, where he built this these honeycomb building blocks, hexagonal building blocks that self-assemble into a hexagonal crystal that has similar dimensions as what I showed you before, a couple microns by a couple microns. And Ned Siemens' group published very nice work in which they designed a building block that looks kind of like a two-layer Rodman-style origami, and we're able to self-assemble this into a rectangular array, again, a couple microns by a couple microns. But I'd like to emphasize that with DNA origami, it's just a couple of idiosyncratic cases where we've been able to succeed to build these very large crystals. But with the single-stranded bricks, it seems like most of the things we try work, and it's just much, much easier to design. You just leave out some strands, and then now you have a new crystal. Thus far, what we've observed is that the DNA brick crystals seem to be more robust than the scaffolded DNA origami crystals. With the origami crystals, we just have a couple of cases where it seems to have worked. With the DNA brick crystal, it's very simple for us to just leave out some of the strands and make a new crystal, and something that's more rigid, higher quality. So hopefully in the future, we can develop methods for improving DNA origami crystals. But in the meantime, we can speculate about why currently the DNA brick crystals are forming better. So we can do the thought experiment that maybe for the DNA origami crystal, you could imagine either preforming the unit cells. You could imagine preforming the unit cells, and then now you mix them together. And the problem is that because the unit cells are so large, it can be very difficult to get reversible assembly. So you make so many contacts with the growing lattice that it's hard to dislodge yourself. And note that this is the same kind of difficulty that plagues macromolecular crystallography, that it becomes very difficult to crystallize large complexes for this reason, among others. Let's contrast that with the DNA brick crystal growth, where the growth is occurring through these very short elements that are only 32 bases long. Because they're so short, it's very easy for them to come in, come out. If there's an error, it has the chance to leave. But because there's many different kinds of bricks, you can still achieve a very complicated unit cell. So it's almost like you can have your cake and eat it too. You can have a very complex unit cell, but it's assembling one subcomponent at a time, so you still get that reversible self-assembly that seems to be critical for robust growth of a crystal. So in conclusion, what we've seen from the lab of Peng Yin over the last year or two, and we also collaborated to help them on this, is a fantastic new method for self-assembling structures that are the size of a ribosome or maybe even larger. We can build them in 2 dimensions. We can build them in 3 dimensions. Right now, what it looks like is there's an especial advantage with these single-stranded bricks in growing periodic structures. And we believe this could have important applications in ranging from molecular electronics and photonics to structural biology.